Hello, and welcome to the AI Research Roundup. I am your host, Alex. Today is May 12th, 2025. We're looking at an ICLR conference paper, ProtComposer, Compositional Protein Structure Generation with 3D Ellipsoids, published a few months ago on January 22nd. This paper offers a new way for AI to design proteins with much better user control. It fine-tunes a specific kind of AI, a flow-matching model, which excels at creating complex structures and uses SE3 residue frames. These frames are precise 3D descriptions of a protein's parts. The main breakthrough in ProtComposer is using 3D ellipsoids. Think of them as user-defined 3D zones to guide the AI in arranging protein substructures like alpha helices in the final design. Alright, so this figure really illustrates how ProtComposer achieves this. On the left, we see the model taking a noisy protein structure and using those user-defined ellipsoids, say for a helix or a sheet, to guide generation. It incorporates this information using a special technique called invariant cross-attention, or ICA. Invariant cross-attention is an SE3 equivariant mechanism, which means it processes 3D rotations and translations of these protein parts consistently. Then, the middle and right parts show these ellipsoids can be statistically generated for novel proteins, or hand-specified for precise user control. So, building on that idea of using ellipsoids, this next figure, figure 2, shows us how these guiding shapes can be extracted from existing protein data. First, you start with an input protein. Then, the method annotates the residues, which are the individual building blocks of the protein, and identifies groups of residues with similar features that are close, specifically within 5 angstroms. An angstrom is a tiny unit of length for atomic distances. Finally, it fits mathematical functions called Gaussians to these groups, defining the 3D ellipsoids. This process is key for automatically generating layouts for redesign or statistical modeling. Okay, so now, figure 3 gives us a visual on how this ellipsoid guidance, using a technique called classifier-free guidance, actually works in practice. We see 2 different target protein layouts, top and bottom. For each layout, the model generates structures with increasing guidance strength. That's the lambda value you see, going from 0, which means no guidance, to 1, representing full guidance. As this lambda increases, the generated proteins, with their blue helices and gold sheets, more closely conform to the target ellipsoids. Metrics like coverage, which is how much of the ellipsoid is filled, and accuracy, how well the structure type matches the target, clearly improve, demonstrating precise user control. Alright, so this table, table 1, really quantifies that control. It compares ProtComposer at different guidance strengths against models like Multiflow and Chroma, and an Oracle ideal. Metrics like coverage, accuracy, and likelihood improve for ProtComposer as lambda increases, meaning generated proteins better match the target ellipsoids. Misplacement and JSD. That's Jensen-Shannon divergence, a measure of how different generated structures are from target distributions, also decrease, showing stronger adherence. ProtComposer clearly approaches Oracle performance with stronger guidance. Next, figure 4 shows how ProtComposer, using its statistical ellipsoid model, compares to other methods. We see Pareto frontiers. These are curves showing the best possible trade-offs between designability, essentially how likely a structure is to be a real foldable protein, and other key traits like diversity, novelty, and one-helicity. Higher one-helicity means less purely helical structures. ProtComposer, in orange, significantly expands these frontiers, generating more diverse, novel, and compositionally varied proteins that remain highly designable, often better reflecting real protein characteristics, indicated by the black PDB dot. Building on that improved diversity, figure 5 gives us a striking visual comparison. On the left, you see random protein samples from Multiflow. The paper notes that often, like in 6 out of 10 cases here, these simplify down to a single alpha helix ellipsoid. In contrast, on the right, ProtComposer's samples clearly exhibit better secondary structure diversity. That's a richer mix of helices and sheets, and more complex ellipsoid compositionality. This really highlights how ProtComposer generates more varied and structurally interesting proteins than the base model. So to wrap up, we've seen how ProtComposer offers remarkable control over generating protein structures by using these 3D ellipsoid layouts. This isn't just about making proteins that fit shapes. It's about significantly boosting their diversity, novelty, and compositional complexity, all while ensuring they remain highly designable, meaning they're likely to be functional. This fine-grained spatial control is achieved, in part, through a special network mechanism called invariant cross-attention, or ICA. Ultimately, ProtComposer presents a powerful new avenue for rational protein design. That's all. For this AI Research Roundup, thanks for tuning in.